All right, we're going to talk a little bit about managing whiteflies. This entire session is on management of whiteflies and managing viruses, whitefly transmitted viruses. We're doing a lot to kind of update everybody on what's going on with this. It's a fairly large project across multiple states, multiple organizations. The part I'm going to cover, we're doing a lot of non-insecticidal work. The part I'm going to cover is insecticides, some of the work I've been doing with that the last couple of years. If you're looking for some miracle coming down the pipeline as far as an insecticide, it's not coming. I'll go ahead and tell you that. I'll make a real short presentation for you there. But we're going to cover some of the work we're doing with that and related work with insecticides. I've been doing these at-plant drench tests for probably 15 years now, primarily monitoring the efficacy of the neonicotinoids and the diamide insecticides. Didn't do the same test I've been doing last year, but it did some of the products that we've looked at. And the bottom line is that really hasn't changed the last 5 years or so. We're getting good efficacy with the soil applications. It's lasting 2 to 3 weeks in most cases. You know, when they first came on the scene, they were lasting 4 weeks, 6 weeks. That's never going to happen again unless we get a new chemistry that's got that kind of activity. Our resistance is not severe, but we've got enough that those products are not going to last that long. But you can see here that we've got, where we're drenching with some of those systemic insecticides, we're establishing a crop, we're keeping it green, and this is yellow squash, which is extremely sensitive to whiteflies, very low populations will give you silver leaf. So where we're suppressing that, we're getting very good control. That's with the direct seeded, the soil application. We continue to look at foliar applications as well. Again, long story short, we can control whitefly with insecticides. It's not cheap, particularly in South Georgia and Tift County and Colquitt County when we've got, you turn over you get a squash plant up with one leaf on it and you turn it over and it's got 100, 200 whitefly adults on it. You're going to have to do a lot of treating to maintain that, to manage those populations, but we do have insecticides that will work. I'm not aware of, we continue to look at resistance. I'm not aware of any severe resistance issues where it's just absolutely not working anymore. So there's really no change there. One of the things you have to keep in mind when you're evaluating what's going on in your field is the vast majority of these products are efficacious primarily on nymphs. So if you're spraying a product that's primarily efficacious on nymphs and you're evaluating on whether or not you're knocking the adults down, you're not going to be happy with it. So you've got to, you've got to evaluate it based on what they're capable of doing. And then as far as managing viruses, whitefly transmitted viruses with insecticides, that is extremely rare that occurs in South Georgia. Our populations, when they appear, are very large. When virus gets into the system, it overwhelms everything very rapidly. So with really insecticides, we can manage whiteflies with insecticides. We cannot manage virus with insecticides. There you're looking at other things such as Dr. Dutta will talk about the work we're doing with row covers, reflective mulches, those kind of things. And the real solution is going to come when our breeders bring in the virus-resistant plants. That's the way you battle viruses and win. Overall on insecticide efficacy on whiteflies, this is the same slide I used last year because, again, nothing's changed. If you are targeting adults, your best products are PQZ and Savanto Prime. Other products such as Acel, Venom, XRL, even if you go down here to what I list as third-tier products, Safina, they've got activity on adults, and in some trials they'll look as good as some of those others. Some trials, Venom will look as good as PQZ and Savanto Prime, but if you look at it across everything and you want to knock down adults, those are the two products we'd primarily use, PQZ and Savanto Prime. Now, overall, our management of whiteflies still relies very heavily on the Group 28s and Group 4s. Those are the foundation of our management programs. The Group 28s, I will point out that Harvanta does not have a lot of whitefly activity. All right, Verimarker XRL, Verimark in the soil, XRL foliar, Coragen in the soil or foliar, still have good activity on whiteflies. And then within the group 4 is Zafonto Prime, Venom, Acel, Ectara, and generics of some of those do very well. Admire Pro, for whatever reason, tends to fail even in the soil application. Foliar applications, it's never been real good on whiteflies. It's great on aphids, but it breaks down too rapidly in sunlight, so it's not great on whiteflies. But that's the foundation of our management, and we absolutely need to rotate those products because we can create resistance. We know that. We had severe resistance to neonicotinoids probably 10 years ago. We had one year where they did absolutely nothing, and we don't want to go back to that. So rotate those. The second tier products, NAC, Courier, primarily growth regulators. NAC tends to be very good on eggs, and then you get a lot of mortality when the pupae try to emerge as adults. Courier's very good on early instar nymphs. Different modes of action than these others, so we want to get those in there where we can in order to maintain susceptibility in a population. Again, we don't want to just rely on these top tier as I list them there. And then third tier products. I know when I say third tier products, people tend to quit listening. But these actually have, some of these, they have good activity. They're just not as consistent. They're not as strong as these others, but there's good activity. I'll show you some data this last year with Safina. It looks pretty good. It's just not at the same level as those others, but they're different modes of action. So for resistance management, where you can use them, we definitely encourage you to work in those third tier products as well into your management programs. Show you what I'm talking about with Safina. This is a test I did this last year. This is just adult whiteflies. As I said, you'll see Zafonto at the bottom. Zafonto gives us very good control of adults, Zafonto Prime. This is Safina. And I can tell you when I was going through this test, it was very clear in every rep there was one plot that was very clean. There was one plot that obviously was cleaner than the others but wasn't quite as good as that first one, and that was Safina. So it's kind of a third tier thing. It's not as good as those others, but it's very good, and you can use it in your rotations. Of course, the growth regulators, Movento and Sinstar, which primarily the active ingredient that's working there is the NAC, nothing on the adults. If you look at immatures, again, Zafonto is the one that drops out, is a little bit better. That's why those are the first tier products. But you can see, again, Safina looked very good, and the Sinstar, which has NAC in it, over time does a very good job of managing. So we've got a lot of chemistries that we can use. We need to rotate them where we can so that we don't lose any chemistries. And the potential loss of chemistries is one of the things that really scares us. So again, overall, we've got kind of the top tier, the foundation, and then work in those others as much as you can if for nothing other than resistance management. Because if we were, if you hammer away with these and you lose one, you put all the pressure on the other one, you're going to lose them all, and that's not going to be a good thing. Other areas related to insecticides where we're doing a lot of work, particularly Dr. Riley's group's done a fair amount of work on resistance monitoring, figuring out how we might be able to do that. That's one of the reasons I continue to do also the same test in some cases year after year after year, is just to see that we're maintaining efficacy of those products. So we're working with resistance monitoring. Dr. Riley's group been doing some work with sublethal effects. I won't really talk about that today, but we will talk about resistance monitoring. Host plant insecticide interactions. Not sure how we're going to use that in the field yet, but that's some work that's been done that's quite interesting. And then I'll talk at the end about some work I've been doing with surfactants this last year. So first of all, resistance monitoring. Again, one of the reasons I do some of these same tests over and over again is to make sure we've got efficacy. I can do that in Tifton fairly easily, but if you start trying to do it across multiple locations, it gets overwhelming very rapidly. So what Dr. Riley's group worked with, particularly Paolo Cremones, Dr. Cremones, is looking at bioassay approach for monitoring for resistance. What they're doing is going out and collecting whiteflies in these tubes. You've got treated cotton plants that you expose those whiteflies to, and you look at mortality at 24 hours. So it gives you a fairly quick estimation of whether or not that population is susceptible. That's something we could do across multiple locations very easily, do it over time much more easily than just repeating these field tests over and over. So hopefully if we can get this worked into our system, we can monitor the resistance much more easily. And it's easy to do it with a lot more chemistries as well. So here's where they've been working with it. They've done the monitoring, which is the bar graphs. That's kind of what the mortality looks like in the bioassay, 24 hours. And then the lines are NIP counts or counts in the field. And you can see that lines up fairly well. So hopefully we can utilize this and do a little more intensive monitoring for insecticide resistance in the future. In conjunction with that, some work that Jermaine is here did as well that's been quite interesting. In conjunction with some of this monitoring, they did follow-up checking for host plant effects on insecticide response after rearing whiteflies in the cage for a generation and after exposing whiteflies to the plant for 48 hours, and then see how they respond to insecticides. Now, what the ideal here is that whiteflies feed on a lot of different plants, and on those different types of plants, they're exposed to different chemicals, phytochemicals in the plant. And the question comes up is, are those chemicals within the plant that are in many cases insecticide type products, they're defense products in the plant, is that triggering something in the whitefly that would cause a different response to insecticides? So if you've got a whitefly on cabbage, is it going to respond differently to product X than that same whitefly on a cucurbit? So that's kind of what they were looking at. And if you look at it here, they did it with cyantroniloprol, XRL, and with Admire, imidacloprid. And you look down here at where they were. Well, first we look at where they just fed for 48 hours. Not a lot of variation there, nothing significant, but obviously a trend for a little bit of a change. But you look where they reared them on a cage for a generation. You got, different as low as, 35% mortality compared to 80% mortality with the same dose of the same insecticide, just against different populations, depending on what they were reared on. So those phytochemicals what it appears is phytochemicals in the host are triggering some resistance mechanisms in the whitefly. And that occurred here with the different, a single population. And here is 3 different field trials they did, again with different hosts, but spraying the same products. And again, you can see that there's different responses depending on the host. So how you actually going to use this in the field at this point, I'm not sure, but it's very interesting information. We're still figuring out what to do. The other thing you'll see is if you look across locations, Mitchell County, Toombs County, and Tift County, you'll see here that say, pick out cotton. Highest mortality in Mitchell County, highest mortality here. Then you get over to Tift County, it's the lowest mortality. So what's going on there? Is that effect of the host they're on, or is it an effect possibly of the host they came from? So host plant is affecting the efficacy. We just got to figure out how, if we can utilize that information, how we utilize it. And the same thing, similar data for imidacloprid, where they fed for 48 hours, where they were reared, and then different locations. No significant differences here, but again, obviously trends and differences among host plants in these different locations. So again, how we utilize this information is still working on, but it's quite interesting, I think. So overall host plant effect conclusion that insecticide dose response is influenced by a host plant, and that influence is greater the longer they're on that plant. So if you've got something coming from cotton and it moves to a cucurbit crop, that initial response is still going to be similar to how it was on cotton. You get a generation in, and that response may change based on the host plant that they've been on. Then the last thing I want to touch on that I really kind of was hesitant to even discuss, I don't want anybody drawing any great conclusions out of this yet, but one of the questions I frequently get, and there is no good answer to, is what's going to happen when I make tank mix all these different products together? When you put in 2 fungicides, 2 insecticides, a pH balancer, a defoamer, all these things. And the answer is we really don't know until you do it, and you've got to test it to find out. But what I was looking at in this case, I picked 4 insecticides that are translaminar. They're systemic if you put them in the soil, translaminar if you put foliar applications, Savano Prime, XRL, Venom, and Coragen. And then I sprayed them alone. I sprayed them in combination with Dynamic, which is a surfactant that should help them penetrate the leaf and is frequently recommended. And then I sprayed them with Bravo Weather Stick. And the chlorthalonil is not the question here, it's the weather stick part. It's the spreader sticker part in the Bravo that is a potential question to look at. And I'll tell you the story behind knowing that that's a big deal, can be, is working with Agrimec for leaf miner control in celery 20 years ago. And we did all the work, looked great. We got a Section 18. Grower started spraying it, failed miserably. And it took us a few weeks, but we finally figured out every time they sprayed, they were spraying with Bravo, and that spreader sticker was sticking the Agrimec to the surface. It was not getting in the leaf. So it wasn't getting to the leaf miners. Similar things could happen with whiteflies. When we first came out with Coragen, we found out that if you're spraying for diamondback moth, you probably don't need a surfactant. If you're spraying for whiteflies, most of the time a penetrating surfactant will help. Why? Because diamondback moth eat the surface of the leaf. You just need it on the leaf. Whiteflies penetrate the leaf and feed inside the leaf, so you've got to get it inside the leaf. So that's why I got interested in looking at this again. Again, don't pull a lot of information, too many final decisions out of this, but this is what happened with the top line in each case is silverleaf where we were not spraying. This plot is for XRL, and you can see there's not a lot of difference there if you put the surfactant or not. And in fact, the lowest one there happens to be numerically is XRL with Bravo. However, if you go over here and look at Savano Prime, this is Savano Prime alone and Savano Prime with Dynamic. That is Savano Prime with the Bravo. So it appears that we very well may have decreased efficacy by that tank mix. And you see a similar thing down here with Coragen. Coragen didn't work as well as these others, but again, where we had Bravo, a little less efficacy. So that's something that we're going to continue to look at and just see if we can tease some things out. But I will tell you, I've done just enough work with surfactants that if you want to know what your surfactant's doing, you've got to look at the pest you're targeting, the crop you're using, and the pesticide involved, and do it with and without that specific surfactant to know whether or not it's helping or hurting or doing nothing. And if you change any of those 4 factors, you can dramatically change the response. You can have the same pest on the same crop with the same surfactant and change the insecticide, and one will help that insecticide, and the same thing may hurt another insecticide. So that's why that's a very difficult question to answer, is you've got to look at that very specific combination in order to have an idea of what the answer is. And I think that's all I had. So if there's any questions, I'll be glad to address them if I can.
Have you done a lot of research on the whiteflies and green beans?
In green beans,
Yes, sir.
I haven't done a lot of work specifically on green beans. We assume that a lot of the information that we're generating primarily off of cucurbits as far as insecticide work is good. Yeah, and some of it, green beans doesn't have the same thing registered. Viruses, I think Dr. Babesh will probably talk about that. We've done a fair amount of screening in the green beans for virus resistance.
I know that the Pitt County and some of the southern parts of Georgia in the fall, we used to have the fly.
Yes, and different varieties respond Some are much more sensitive to whiteflies than others. You end up with bleach pods. So that's, yes, that's a big issue in green beans. And some of the viruses are very severe in green beans in some years. Yes, and that's the virus, the whole virus issue in green beans, they've got some varieties now, and I think Bhavesh will talk about that when he comes up.
All right, for our next talk, we'll have Dr. Ted McAvoy, our UGA vegetable agronomist. Thank you, Ted.
Thank you, Will. So my talk's going to be on host resistance in cucurbits to whitefly feeding and whitefly transmitted viruses. We tested, again, for the second year in a row, several crops, species, and varieties. So this is not a comprehensive list, but basically we're getting different virus symptoms. We have multiple viruses and multiple virus symptoms. So in my talk, I just characterize virus or no virus with incidence and severity, but this is my PhD student's work, and in the lab she's quantifying and identifying those viruses and the amount of virus in all these plants. And the symptoms they're going to cause are crumpling, yellowing, or death in the case of vine decline. All right, so we had 5 different crops, several different species, Pepo, Sativus, Maxima, and Machada. And we had as many as 10 varieties per crop. We did this work last year, but we included all the same varieties as last year, but we also added more based on the literature of what performed well, what susceptible checks, as well as new varieties that have never been tested. Most of them were straight neck for yellow squash, but we did have crook neck. For zucchini, we had some gray in with the typical green zucchini. In the cucumber, we had a processor pickling variety, slicers, and then a greenhouse variety. For the winter squash, we had acorn squash, kabocha Hubbard, calabasa, butternut. And the reason we added more butternut is because the first year we found good resistance, but we found a variety that wasn't well adapted to Georgia, so we wanted to expand that screening. And in pumpkin, we had the Pepos, Spartan, Secretariat Zeus, and Big Loretta, and then we added Orange Bulldog, which is a Machada, and you'll see it looks a little different. I don't know what I'm doing. Okay. So I mentioned we did a virus screening, but we're also, we did whitefly counts and nymph counts. We're not presenting that, but we got silvering from the feeding as well as virus. So we're starting off with zucchini. We did get differences in silvering in zucchini. You can see the left versus the right with the white. Now, zucchini has natural flecking, but this pure white is silvering from whitefly feeding. And there was differences between varieties. Like I mentioned, the typical virus symptoms are crumpling, yellowing, and death. We saw yellowing and crumpling in zucchini. And when I show the data, you'll see zucchini is much more tolerant than yellow squash. So for summer squash, zucchini is your better choice. So for all these slides, we have count and weight. And they're always going to be in order by the yields. So Renegade was the outstanding variety here this year. It was a new variety that we hadn't tested. But other greens that did well were Everglade and Fortress, which did well last year as well. So that's 2 years in a row. So those varieties are standing up. You can see also the gray varieties, the Hurricane and the Rocchio, did very well. And all these did better than the spineless varieties. People like the spineless varieties because they don't itch when you pick them and they don't scratch the fruit. But it seems like they don't perform as well. I'll also point out in the weight, these are more eggplant shaped, as you'll see, so they had more weight, but they didn't have more counts, excuse me. So on all these charts, I have a stop sign where green is good, yellow's okay, and red is bad. We have, in this case, the lower number is better for number of virus fruit, the number of virus weight, so count and weight for virus symptoms that were unmarketable. And then we also did unmarketable for other reasons, which were not virus related, but were just adaptability in Georgia. So you can see a lot of zucchini had low virus numbers. Again, this Renegade, top performer, across the board, no virus fruit. Same thing with the Brocchio and the Fortress. As far as the width, I put it in red, but it's just different. It's not, as I mentioned, these two are wider, where these other ones are skinny and cylindrical. As far as the spineless varieties, spineless perfection was the worst, and out of the spineless varieties, spineless supreme is the best. So now, silvering, virus, and downy mildew. I'm going to focus on the silvering and the virus, but you could see the results for everything. So the silvering is feeding. Again, these numbers, the lower the better. The average is because we did weekly ratings, so this is the average of all the ratings put together. So this is a 1 to 5 rating percentage, and this is a 0-4 rating. Again, Renegade across the board. It really did good. I hope it does good again next year. Brocchio, as we mentioned, had less virus fruit, also has less virus incidence and severity. So if you want a gray zucchini, there you go. Spineless perfection, again, very bad. So this is what I was talking about, healthy fruit versus virus fruit that are unmarketable in gray and green zucchini. And then this is marketable or unmarketable based on shape with pollination and fruit set. And this is the Renegade, which was the top performer. This clicker is giving me all kinds of fits. And that's the gray zucchini I mentioned. They weighed more because they were fatter. We picked them all the same 3 times a week for 3 weeks with 9 total harvests. That's what all our yields are for our zucchini and our squash. And the Hurricane's the bigger one out of the 2. And this one had less virus. All right, so now we're going into yellow squash. As you'll see, it doesn't perform as well as zucchini is. As far as summer squash, again, silvering and virus. The virus is more pronounced. The crumple leaf attacks the growing point, and that really prevents growth. You'll just have stunted plants that don't do anything once they're infected. So this is what the squash look at the end. You can see more crumpling, more yellowing. They're under deathbed. The fruits aren't harvestable towards the end. But we do have differences in varieties, and I know zucchini is not an alternative to yellow squash. So if you want yellow squash, some perform better than others. So again, fruit and weight, count and weight. Cougar, lioness, multi-pig, and laser were the best for count and weight. And these did better than Gentry, Fortune, and Gold Star. And as you'll see, the Gentry at the end had 100% virus infection. So that's a susceptible check, a known susceptible check. So again, these are just in order of the yields. But again, Gentry, very susceptible, and we have some that are outperforming. Here's our top performers. I don't know if the battery's dying or what's going on. Okay. Stand over here. So the Cougar, Lioness, and Laser didn't silver as much. The Lioness and the Grand Prize had less virus incidence. The Lioness and the Laser had less virus severity. And I'm just going to show pictures of healthy fruit and virus fruit, as well as misshapen fruits that we counted here. So next crop is cucumber, and cucumber doesn't get crumple leaf symptoms. It gets the yellowing symptoms, and because of this, cucumber is a good crop. You know, I'm sure the virus does reduce yields, but not as severely as the crumple leaf virus. Again, here's, you know, up until harvest, you look good. When you start harvesting, you know, you start collapsing. But we were able to harvest for 3 weeks. Here, remember the Supremo was a processing pickling, so you had more small fruit. So that one did well. It's almost unfair. But also the Dasher, Diamondback, and Bristol. You can see the Bristol had the highest weight. These were all better than the Tasty Green, which is greenhouse variety, and they were also better than the Speedway, Brickyard, and Raceway. So, you know, we typically do slicers, but if somebody wanted to grow processing did good across the board. And the Bristol, the one that had the most weight, that did good across the board. So that's something I think is a good option. You know, Dasher 2 is a standard, Diamondback's a standard. So we have options there. I would recommend growing cucumbers in the fall if you're going to grow cucurbits. As far as the virus incidence, the ones I highlighted had less incidence, and in general, you know, kind of tied. I put it in the middle, but kind of tied. Severity as well. This is that greenhouse variety. It was very long. It hit the ground and curved, so that's why it was so affected, but marketable and unmarketable shapes. So now we're getting to winter squash. So winter squash is typically not grown in Georgia. You know, definitely the yellow squash, the zucchini, and the cucumbers, but we're looking for alternative crops as well. So we did winter squash and pumpkins. And there's the most variability here, and I think maybe some opportunity as well. So you can see, depending on the species and crop, silvering and intense virus. And you can see the differences in silvering across the field. So as far as count, all the butternuts had the highest counts. The acorn squash had the least. That trend continued for weight, but here the Genesis by far had the most weight, and as you'll see, that was the biggest. The Ceres and Waltham were very small, and the Ultra was very skinny. So I would recommend butternut squash. You know, it seemed to do good for us, and that's an alternative. The Genesis, which I mentioned, that had the highest count and weight, again, across the board for low virus fruit and marketability. Remember I told you the Ceres and Waltham were small, and this one was real skinny, is why they were unmarketable, not due to virus, not well adapted to Georgia. You know, we didn't get a clear trend. You know, some of the ones that didn't silver were small. So I think there's an opportunity to explore more butternuts. This is the Genesis. This is the one that did well. These ones were small, and these ones were really skinny. The Atlas last year was our best performer, and it has a beautiful shape. You know, I like that blocky shape. But what we're seeing is it tends to split. So right now, Genesis looks like to be the best performer. And then virtually all our acorn squash look like that. and this is the Hubbard. And lastly is pumpkin. In short, I do not recommend growing pumpkin. I know people might want to grow it for a roadside stand or U-pick. So there were differences in varieties, and we'll show you which one did the best. This was also a longer season, and we got collapse at the end. Again, we found in watermelon that we had the vine decline, and we're going to test all these species for vine decline as well. In general, I think it seems like the shorter matured crops, could get in and out quicker and possibly avoid some of these problems. Again, more yellowing, vine decline, silvering differences. So just like last year, the highest yielder was Spartan for count and weight. But again, high number of unmarketable across the board. I would probably not do it commercially, more of a U-pick type situation if you really were dead set on it. The typical pumpkins tended not to silver compared to the orange bulldog, but the orange bulldog had less virus severity. It's a machata. And this is what they look like. There's the Spartan, the best performer. So I'll just summarize. As far as summer squash, zucchini outperformed yellow squash. For green zucchini, Renegade was the best, followed by Everglade and Fortress, and that was 2 years in a row for those ones. Renegade we only tested once. The specialty gray types did really well, Hurricane and Rocchio. The Hurricane was bigger, the Rocchio had better virus tolerance, and the Spilers performed poorly. But out of those, the Supreme was the better performer. Out of yellow squash, Cougar, Lioness, Multipick, and Laser. Out of the cucumber, the slicers that we primarily do is Bristol did the best, followed by Dasher and Diamondback. And for pickling, the Supremo did well. And for our winter squash, Butternut did best across the board, and out of the Butternut, the Genesis performed the best. And out of the pumpkin, didn't do too well, but Spartan for 2 years in a row has been our best performer. Now I'd just like to acknowledge everyone, USDA, ARS, for funding this project. We're very appreciative. This is my graduate student's project, Nirmala, and her committee, but everyone in the room, Dr. Baggs on the committee, but Dr. McGregor's helped. Luan is taking drone images, so we've got a whole team that's contributing here. My research technician, my postdoc, Manisha Kumari, and then also Sudeep's postdoc, Manish Kumar, and all my graduate students sitting here in the second row. And with that, I'd like to answer any questions.
Thank you, Dr. McAvoy. For the next talk, we'll have Dr. Dutta, the UGA vegetable pathologist.
Well, hello. Well, good afternoon. Before I start, I'd like to give 1 minute to Dr. Rebecca Melanson from Mississippi State. She has something to talk about whitefly and whitefly transmitted viruses.
Oh, Whitefly stuff, not this one? Okay, well, I'll do two things since I have a minute, and I'll make it short. So I am co-chair, along with Dr. Wintermantle, of the Emerging Viruses and Cucurbits Working Group, and we have a booth. Obviously, through this session, you see that whiteflies and their viruses that they transmit are very important to cucurbit crops as well as other vegetables. You heard Dr. Dutta speak earlier this morning. One of the main missions of our group is to try and educate stakeholders and growers about these viruses. And so we have a table in the rotunda as well as some postcards on the back table here about just short educational postcards and publications, fact sheets about some of these viruses, the symptoms, how they can be detected, how to manage those. So feel free to stop by, take those, visit our website. And then the other thing that I want to mention, I will put these postcards in the back too. We have a new app, the Southeastern Vegetable Extension Workers Group, that is group that produces the Southeast Vegetable Crop Handbook that Dr. Dutta works on as well. We are starting a My IPM for Vegetables app, and it currently has information about cucurbits and tomatoes. And so that is available and free to download, and it has information in there about insects and diseases of those two crops. And then we plan to add more in the future. And if you are here tomorrow around 2 o'clock, Aaron Ure will be giving more information about that app. So I will put these in on the back table as well. Thank you.
Thank you, Rebecca. Before I start, again, I have a couple of introductions. We have with us our new vegetable pathologist, research pathologist, and she is also a distinguished research professor, Dr. Myung Tian. She just joined our department last week. She's coming from University of Hawaii, and she is a molecular geneticist who's going to help us with Phytophthora capsicum. And we also have Dr. Luan, our specialist, and and he joined last year, but this is his first year as in this conference. All right, coming back to my topic. So I'm going to tell you, I'm not going to tell you all the data and this and that stuff. I'm going to give you gist of some of the trials which we have been doing, some of the work which we have learned over the years. Before I start, I'd like to acknowledge the funding source is USDA cooperative funding that came from USDA. Really appreciative of those funding. I also like to acknowledge some of our UGA vegetable virus team that includes Dr. Babu Srinivasan, Dr. Sudeep Bagh, Dr. Ted McAvoy, Dr. Cecilia McGregor, and Dr. Stormy Spark. Without their help, some of this stuff which I'm going to talk today will not be able to do. I'd like to also acknowledge our team of excellent vegetable county extension agents who helped us throughout the journey in producing some of this data. So thank you all. So my talk is on spread and management of whitefly transmitted virus complex, okay, in vegetable crops. So there are two vegetable crops I'm going to talk today. One, I'm going to spend some time on snap beans. And the vegetable virus complex we see in snap bean include Cedar golden mosaic virus and Cucurbit leaf crumple virus. Okay, there's two viruses. But in Georgia, what we have seen, the main driver of the viral symptoms in snap beans is Cedar golden mosaic virus. So if I talk about snap beans, it's mainly Cedar golden mosaic virus. But second half of my talk would be on cucurbit, and their whitefly transmitted virus complex includes, importantly, three viruses. They occur sometime together in complexes, either 3 or in 2. The three viruses include Cucurbit leaf crumple virus, Cucurbit yellow stunting disorder virus, and Cucurbit CCYV. So you're looking at the 2-month-old healthy snap bean plant. This is how it should look, right? This is another 2-month-old Cedar golden mosaic virus infected plant. It did not grow more than 7 inches from the ground. This is Cedar golden mosaic virus can do to snap beans. In 2017, we had 80% crop loss in fall-grown snap beans in Georgia. 80% crop loss. That accounts to close to $7 to $8 million. It can go beyond. It can go up to $50 million if you include all the input costs and so forth. So that much of damage we saw in 2017. This virus has a potential to do this kind of damage. We have done a lot of varietal screen and a lot of PI line screen, and I have all the data. And usually every year through our extension system, I give away all those rankings of different commercial varieties of snap beans which growers can think in growing their field during fall. Those informations are provided through the extension system. But those individuals who need those information who are outside the extension system, outside Georgia, please contact me. Okay, I'll have the list. Okay, switching gears. In this particular talk, I'll spend some time on where Cedar golden mosaic virus survive in our natural system in Georgia. So it survives in different weed hosts. One of the important weed hosts is prickly cedar. It's a Malvaceae family member. It is quite common in Georgia, but its epidemiological studies, there are no prevalence studies has been done to demonstrate the prevalence of these infected prickly in commercial snap bean fields and how important they are in transmitting the virus from these weeds to the snap beans. So we asked two questions. First, how prevalent is prickly in snap bean fields? And second, can they be a source of inoculum for this subsequent snap bean crop? So this is our hypothesis, prickly whitefly can feed on them, can transmit the virus. You'll see the infected prickly and from there, again, whitefly can acquire and transmit it to the to the healthy snap bean plant. This is our hypothesis. So this part of the work was done by my PhD student, doctor. Now he already finished his PhD, Dr. Clarence Godard. And this particular work has been accepted with some revisions in plant disease. It should come out in a couple of months or 3 months. So snap beans, so what our target is, what is the source of inoculum for snap bean viruses, particularly Sida, in the fall? So we sampled the weeds in spring and summer, particularly in spring. So we looked at 17 commercial snap bean fields in 3 counties, Worth, Taft, and Sumter. Okay? What we found, except for one location, all locations had prickly Sida. Prickly Sida is quite perennial weed. Okay? And when we looked at if those prickly Sida had symptoms or no symptoms, these are the symptoms we are talking about, mosaic symptoms. We found nearly 134 weeds sampled across these locations. Only none of them had any symptoms, but 3 of those asymptomatic samples was positive for Sida golden mosaic virus. So most of the asymptomatic plants were truly asymptomatic. Only few had Sida golden mosaic virus. But there are few locations that also had symptomatic prickly Sida. They had typical virus symptoms. And when we tested for the Sida golden mosaic virus, close to 89% of them were positive for Sida. So what does this suggest? First, prickly Sida can be prevalent in snap bean fields during fallow season, and also those symptomatic prickly Sida can be infected with Sida golden mosaic virus. Okay, so based on, now we have 17 locations. Out of 17 locations, 9 locations had symptomatic prickly Sida. We designed a study to assess the spread of the virus from prickly Sida to snap bean field. So this is an example of a snap bean field where prickly Sida was found on this portion, infected symptomatic prickly Sida. And on this portion, there were no prickly Sida. So what we decided, we designed a study where we set a field study where we grew snap beans here and we grew snap beans here. The only difference here is this particular location had a prickly Sida. This particular location did not have a symptomatic prickly Sida or no prickly Sida. And if you look at the difference, the distance between these two is close to half kilometer, 475 meters. Okay, I'm not showing all the data for all the 4 farms, but I'm showing you some of the snapshots of the couple of farms. This is the one farm. We're looking at these are the plots over the period of plantings, 21 days after planting to 42 days after planting, and you're looking at the plots without symptomatic prickly Sida. In those plots, we did not see any virus infection on snap beans when it was planted. Whereas the snap beans that were planted close to the prickly Sida, infected prickly Sida, we had virus incidence starting from 28 days after planting, and then it moved up to 42 days after planting. So this shows there's a local source of inoculum that can drive the infection. Another field. Again, prickly Sida was present on this side of the field, and the infection started early, and there was a lot of infection. But there are also plots, these are the snap bean plots, that did not have any prickly Sida, but we still saw some infection. So what does it suggest? It suggests the whitefly can also disperse from vast distances And virulophilus whitefly can also disperse from vast distances and can transmit the virus to snap bean fields, to these snap bean plants, irrespective of presence or absence of prickly cedar. But if you have a prickly cedar or local source of inoculum, your chances of severe virus infection in snap beans is much more steeper. This is what it says. Let's look at some of the data. So we looked at the 4 commercial farms. You're looking at the disease incidence. The red ones are the disease incidence in snap beans with prickly cedar, and this one without prickly cedar. I can see in every occasion, the snap beans that were planted close to infected prickly cedar had a more disease incidence compared to those that did not. If you look at the AUDPC, it also followed the similar trend. High AUDPC in snap beans that were planted close to prickly cedar compared to those that were not. So what is the summary of this particular study? Prickly cedar can serve as a reservoir for this virus. Prickly cedar can serve as a green bridge of the virus between 2 snap bean crops. And prickly cedar is also a very good host for the whitefly too. Whitefly can also, I did not show the whitefly population data, but whitefly can also thrive on this prickly cedar. So what is the take-home message here? If you are a snap bean grower or if you're a consultant, make sure you follow very good weed management around the field and within the field. Especially if you see prickly cedar, get them off. So, any questions on this portion of the study? Okay, this shows the impact of natural reservoir and the green bridge in persistence of the viruses within the system. So I'm switching gears now. So now I'm going to move on to a different crop. So squash, which can be heavily infected with the whitefly transmitted virus complexes, and under severe conditions we can see losses as high as 60 to 70%. So I'm not going to talk about these because my previous speakers they have already spoken about these things. But again, just to remind, when you talk about the whitefly transmitted virus complexes here in squash, we're talking about 3 main viruses: Cucurbit leaf crumple virus, CYSDV, and CCYV. Okay. So this is the rationale why we did this study. For example, in squash, we do not have a good commercial variety that has good level of resistance or tolerance against these virus complexes. Second, you cannot rely on insecticide. Insecticide can reduce the level of infestation, whitefly infestation, but you just need few virulophilus whitefly to transmit the virus. So it will reduce the whitefly population, but it will not reduce the virus incidence and severity. So the other thing is that, that there's also risk of insecticide resistance and so forth. So we will always look out for additional pest management tactics, and integrated pest management is the key in this case. So what we did, we took a different approach. Okay, we relied on an already well-established risk management system in peanut. How many of you heard about peanut RX against tomato spotted wilt virus? It's well studied. It took 30 years of research to get the peanut RX. What peanut RX does, it gives you the list of variables. Okay? A grower can choose these variables. Each variable will come up with a risk. Okay? Depending on what the grower wants to do, he can choose those risk points and total the risk point, and he can predict the risk of the tomato spotted wilt virus. For example, what peanut variety he'll be using, what planting date, what plant population, what row pattern, what tillage, and what herbicide or insecticide. Depending on that, depending on those variables, you can predict a risk. Now we asked a similar question in squash. Depending on different management practices, can we assign a risk variable to those management practices and then predict a risk of whitefly transmitted virus complex in squash? So we started looking at different variables. So we did multi-years of studies. So the data which I'm going to show you today is an outcome of close to 25 different field experiments run across for 3 years. So this is a trial. We looked at the first variable, which is squash genotype, a cucurbit genotype. What we found that, and Ted also mentioned, the winter squash is much more tolerant than the summer squash. So you're looking at, these are the summer squash, so more virus in summer squash across the years compared to the winter squash. So it's one variable. So if you are growing a crop, your fall squash, and you stick to this yellow squash, you are at more risk of the virus incidence. Okay, what about planting date? So we looked at the planting date from August 17th to October 15th. Before going to the planting, let me ask you this question. When is the peak of whitefly population that occurred in our region, in Tift, Colquitt, and Worth County? Do you know what is the peak? When is the duration of the peak? It is from August 15th to September 15th. That's the peak. It can move a week here and there, but more or less, that's the peak. So if you grow our crop, susceptible crop, within that peak, you are at more risk of a virus incidence. So here, this is what it shows. Here, squash and zucchini both were planted on August 17th, August 31st, September 15th, September 30th, and October 15th. Where do you see more virus incidence? What dates? Between August 17th to September 15th. Look at when you grow crops beyond September 30th. You can ask me a question, what happens if you grow crop before? Well, in this particular trial, we did not do it, but in subsequent trials, we did it. In this particular trial, we looked at insecticide and planting date interaction. This trial was actually done by Dr. Stormy Sparks in collaboration with my student. Here in 2020, they looked at insecticides and planting date. They had 10 planting dates and this insecticide program. In 2021-22, they had 8 planting dates and so forth. Don't worry about those minutiae. I'm going to give you the gist. In 2020, the major gist is insecticide will not reduce the virus incidence. It can reduce the whitefly population, but it doesn't have any impact on virus incidences. But planting date does have. So look at here. Here, planting was done from June 29th to August 31st. When do you see more virus incidence? From August 3rd to August 10th. This is the period when you see more virus incidence. But if you want to grow crop before August 3rd or before August 1st week, you are at lesser risk. In 2021, we did not have a lot of whitefly population. There were a lot of weather events that actually reduced the virus and the whitefly population. So we did see some trends, but it was not too great. Look at 2022. Again, same story. When did the virus peak happen? August 15th to September 15th duration. So planting date has a big impact, has an impact in our region. Now, when we talk about squash RX, it is very restrictive to our region, to 3-4 southern counties, okay? You cannot take this and run away and do similar risk index in Michigan or somewhere else, okay? All right, so what did we learn? Plant genotype is important, planting dates are important, insecticide doesn't work at all, and those 3 things. Let's look at other things. The other variables we looked at are row covers and reflective mulch. So in one experiment, we looked at a reflective mulch and that with a row cover, single row cover. This is single row cover. We looked at reflective mulch without row cover, white plastic, single row cover, and white plastic, no row cover. Okay? In another experiment, we also had the similar thing, but instead of row cover, we used a 6-row floating cover, which is strawberry covers. Look at these things. What you're looking at is the fate of the squash plant after 24 days of planting. Now, one thing which I want to mention, the row covers were put for just 10 days. First 10 days of transplanting. After first 10 days, the row covers were taken off because you need to help with the pollination and so forth. So this is 24 days after planting. This is a white plastic squash. You can see how bad it looks. Look at reflective mulch, but with no row cover. It is better than this one, but not too great. But look at these row covers. So this is white plastic with row cover, UV reflective mulch with row cover. So it seems like row cover is the main driver of virus incidence reduction of virus incidence compared to the plastic mulch. Plastic mulch will give you some advantage, but not a whole lot. UV mulch can give you some level of advantage, but not a whole lot. If I were a grower, if I know there's a risk of growing this crop in the fall and I'm predicting the risk, I would rather go with the row covers for first 10 days. Okay, let's look at some of the data. So this is the disease incidence. Can you predict what it is? What kind of treatment? White plastic, no row cover. And these are row covers. Look at the disease. We did not see much virus here. This is when you compare two different plastic mulch and look at and compare the yield, we had more yield with UV reflective mulch. As I mentioned, UV reflective mulch will help something compared to white mulch. But when we have a row cover, the yields are drastic. So you have more yield with covers, row covers compared to the no row covers. And this was consistent across the years. Again, row covers is the main driver of a reduction of virus incidence compared to the plastic mulch, UV mulch. Okay, this is the fifth variable we looked at, type of seeding material. Are you starting a crop with direct seeding or starting a crop with seedlings? We saw if you start a crop with a seed, direct seeding, you have more virus compared to the seedling. Why is it so? Because you are exposing your crop right at the start with the whitefly. As a result, you have more viruses to start on when you do the seeding compared to the seedling stage. Come on. So now we have all this information. How we can rank all these variables? How we know what variables or what predictor variables are of more importance? We used some fancy statistical analysis. My student did that. He used random forest analysis, and he found that use of row covers is the top number one predictor of your risk, okay? Compared to date of planting, mulch type, squash type, and planting material. And depending on that one, we ranked these predictor variables, and we assigned risk points to each variable. Now I'm going to show you how we did that. For example, planting date. So if you plant before August 11th, you will be assigned at a low risk point compared to if you plant between August 25th to August 11th to 25th or August 26th to August 15th. So you will be at higher risk. Row covers. If you choose not to use row covers, you will be at a higher risk compared to if you choose using row cover. Squash type. If you're using zucchini, you're at a lower risk compared to you're using squash, yellow squash. Planting material, you'll be at a lower risk if you use seedling compared to seeds. Mulch type, you'll be at a lower risk if you use a reflective mulch compared to the white plastic. So growers can use these or combination of these or all of these and add these risk points and come up and predict the risk. Okay? So depending on that, if you are combining all the tactics and your risk points falls between 45 and 75, then you're at lower risk. And if you fall between 120 to 150, you're at higher risk. Now, it's a time for validation. So what we did, we created scenario where we grew squash at low risk scenario, high risk scenario, and moderate risk scenario. What is a high risk scenario? We planted this crop in September at the peak of whitefly population on white plastic without any row cover. Our low risk was yellow squash planted on a UV reflective mulch with a row cover. We saw a positive correlation between the risk points and ADPC value, suggesting those plots we predicted as a high risk were indeed had a higher standard ADPC compared to those plots we assigned as a low risk. Come on. Okay. We also looked at the marketable fruits. We looked the fruits from the high-risk plots were quite low compared to the fruits from the low-risk plots. And this is all significant. We did the validation for consecutive 2 years. This is the cool stuff. We went to 22 different commercial squash growers' field. Okay? We asked them what are the variables, what are the practices that he is using. And based on that, we came up with risks. Okay? And then we also tracked the virus incidence and severity in those 22 different commercial fields. And when we plotted them with the risk and the observed viruses, it was significant. It means the risk which we predicted based on what the growers have done was significant. It means we are on the right track in predicting the risk of the whitefly transmitted viruses in squash. Moving on. This is an example of a grower, okay, who was at higher risk of a whitefly transmitted virus complex. Look at the variables he was using. He used yellow squash, which is highly susceptible. He used a planting date of September 2, which is within the period of August 15 to September 15 of whitefly peak. He used a white plastic mulch. This black plastic was painted white. He did not use row cover, and his planting material was seedling. We found that he was at a cumulative risk of 145, which is higher risk based on our assessment. We went back, we looked at the virus incidence, also we looked at the yield reduction. He had 40% yield reduction at the end of the season. So this shows some kind of validation. Although we are on the right track, we also have to do further validations, but this shows we are on the right track. And this is the first attempt where we have used risk index to predict risk based on the predictor variables, different practices. And this is also the first instance where random forest analysis with statistical analysis have been used to predict risk. So with this, I'd like to conclude my talk, and I'll be happy to take questions. Okay, if you are in Georgia, southern Georgia, and you're talking about the fall, and talking about the 3 or 4 counties, southern Georgia counties, there we'll see highest diversity of vegetable crops during the fall. We grow bunch of solanaceous crops, bunch of cucurbitous crops, bunch of brassica crops, and also cotton is also there, which can also be a source of whitefly infestation. So we have a system where the virus and the whitefly sustain over the period of time. Now, if you use a row cover, okay, the row cover helps. The row cover is only for first 10 days. After that, you remove the row cover. Those first 10 days of virus or whitefly-free period is enough to reduce the virus incidence over this season.
So your take-home message is row cover is really, helpful for...
Row cover, look, you have to first see what is the risk of this particular fall for virus and virus incidence and whitefly populations. It will drive your decision to use row cover or not use row cover. In the moderate level of whitefly infestation, low level of whitefly infestation, I would not. But if you are a grower, you want to use yellow squash, you want to grow yellow squash because of the market, because market drives everything, our decision. Okay, not the pathologist will drive the decision, market. So if the market says yellow squash is more beneficial to use because market price is high, go for it, but use the best possible practices that will reduce your virus incidence. Yes, Bill.
I like your explanation there. One question I would add is, how would you work that into the
That's a very, very important question. Some years will be cost effective, some years are not. If you have let's say, if the market price is high and you are choosing with the yellow squash, go for those. But in years, when you predict there will be a lower market price, less virus incidence, less whitefly population, then you can go without it. But in certain situations, you have a high whitefly population, high market price. In that case, it's your decision what you want to do. But again, extensive economic analysis need to be done here. We have not involved economics here. That needs to be done. Yes?
In the low-risk scenario, are you still getting an acceptable level of disease?
Yes, yes. I have shown you the marketable yield and the number of fruits. Yes, we do.
I was wondering what your row covers here are. Those are plastic nets.
No, those are breathable plastic nets. It's available.
Are they reusable every season?
Yes, you can reuse it. There are some commercial growers who use row covers.
That could make that cost effective. And
Yes, yes. As long as you don't have any tear or breakage in your row covers. You don't want to have a tiered row cover where whitefly can enter and it will, and then your the tactics will be implicated. Yes, Luhan.
So you said like 40% yield reduction, right?
Yes.
I mean, it's risk situation.
Yeah, yeah, in one of the cases.
Yeah. How much is the class on?
I agree. Well, in that particular situation, the 40% was based on his baseline. Okay? Okay. So it was, he did not use any row cover, he did not do anything. But if, in our case, if you want to repeat the study under designed risks, assigned risk, I would include extensive economic analysis. And it has to be done on a larger scale. Okay, cannot do at experimental field scale. Yes, Amol. Yes. No, that was a showcase, but we have the data for all 22 farmers. Some of them were high risk, some were they were at a low risk, some of them are moderate risk. Only few were at low risk.
Was there consistency in the findings, like did you find
And yes, if you remember, I show a regression line that correlated with the observed area under disease progress curve in those 22 farms with the risk we predicted based what they have used. So yes. Questions? Well, thank you. Thank you for listening.
Thank you, Dr. Dutta. For our next talk, our speaker will be Dr. George Mabata with the Fort Valley State University.
Good afternoon, everybody. I did this work with Dr. Yimping, who was my postdoc. She left a couple of, about 6 weeks ago, yeah, a couple of months ago. And then Dr. Alvin Simmons. This study was funded by USDA ARS, and we are really grateful for the funds. We investigated susceptibilities of squash, yellow squash cultivars and the zucchini to the sweet potato whitefly. We know that the whitefly causes a lot of damage to vegetables, and it's been estimated that about losses of about $161 million were incurred due to infestation by the whitefly in Georgia alone in 2017. And this particular pest, it causes yellowing. It causes it transmits several plant vegetable viruses, and also does discoloration of the leaves, preventing photosynthesis. So the losses are really very major, and it could result in total crop collapse in some seasons. The best management for the whitefly, most of the time, most people, it's a lot more convenient to use pesticides. But we know that the whitefly has become resistant to several pesticide classes, and alternative methods are really needed, particularly environmentally friendly methods and also biological control methods are needed, to mitigate the impact of wildfly on vegetable production. Host plant resistance is one of the IPM tools that is employed in the management of crop insect pests. And with the whitefly also, some varieties have been noted to be less susceptible than others to the pest. With some squash cultivars are also known to be resistant to the whitefly. We investigated about 18 squash cultivars. That's among the squash cultivars we had 9 yellow squash and then 9 zucchini varieties. So our objective was to determine the objective were twofold: determine the different susceptibilities to the cultivars, and also to evaluate yield of the cultivars. Our design method, we carried out our study in 3 seasons. We had a 2021 summer season, then fall 2021 and 2022. Our studies involved a randomized complete block design and 3 blocks. Each of the blocks had 19 plots, or 18, depending on the number of varieties that we are cultivated. For summer 2021, when we cultivated the 18 varieties, we had 18 plots in each of the blocks, and the varieties were assigned randomly to the plots. For fall 2021 and 2022, we had 19 cultivars, so we had 19 plots, which we are as well assigned randomly, to the varieties. Data analysis we used was generalized linear mixed model. And the data we collected were, whitefly adult counts and eggs and the nymph counts. Then we also evaluated the yield of the various squash varieties. These are the yellow squash varieties that we investigated in summer 2021. And these are the zucchini varieties that we evaluated in the same season. And in 2021 and 2022, we added we had an additional cultivar of yellow squash, the Gourmet Gold hybrid. These experiments were carried out on the university farm. These are our results for summer 2021. These are Miam adults, Miam 1 adults, that's whitefly adults. The highest counts were found in week 1 and week 2 of our sampling. In fact, our sampling started 25 days post emergence of the plant. So we found the highest number of highest incidents or counts of the adults. Then they tapered off. The lowest was in week 5, and we assumed these other smaller peaks to be peak populations. That's generations of the adults. So. These are the adult counts on the different varieties. The number of the adults was lowest in golden, in the variety, golden goose hybrid, and the black beauty also is low in golden glory, and the green eclipse. And the highest number was in peak and peak. These are attracted most of the, it attracted more adults than the rest, followed by this fortune. These are egg counts. The eggs, we found that the number of eggs were highest on the 3rd week of sampling. And the highest number of eggs trailed the peak populations of the adults. Now, the number of Whitefly eggs were lowest in Lioness, Fortune, and the Ampic 8400 cultivars. They were also low in gray and the shorting hybrid. And these are the yields, the yield of yellow squash. The yield was highest on Lioness, Fortune, and the variety Amber Peak. Yield of zucchini varieties, that's for summer 2021, was highest in Golden Glory. These are environmental conditions, during fall 2021. The highest temperature, the range of temperature was between 11.82 and 21.67 degrees Celsius. Humidity ranged between 70.28 and 91.80 percentage. And then rainfall was between 0.04 and 7.98 millimeters. These are results of adults. Again, the adults were highest on week 1 and 2, and lowest week 5 and 6. These are egg counts. Egg counts here was highest on week 1. This tended to correspond with the highest number of adults. And this is, among our sampling seasons, this is the only time we saw the number of eggs, the egg counts corresponding to the number of adult counts. Usually what we found in several of the seasons we've studied is that the egg laying tended to trail or follow behind adult peaks. eggs were number of eggs the number of eggs was lowest in lioness, fortune, and black beauty and gray. These have a very low number of eggs also. Right through here, respect, golden glory, Ford hook, and the short-in hybrid. These all had a very low number of eggs. And one thing we noted is that if some varieties would have high number, will attract adults, but there will be very low eggs on them, which may imply some level of antibiotics or some kind of repellent, something preventing them from laying eggs on those varieties. These are environmental conditions for 2022, for fall 2022. The temperature range between 7.54 and 19.12 degrees Celsius, while relative humidity range between 77.90 and 57.8, between 57.80 and 77.90%. Then precipitation was quite low, but the fourth week of sampling, we had a fairly good precipitation. This is a 6.50 millimeter. This is a summary of Miam adults for fall 2022 on the different varieties. The highest number of adults we also found week 1, this average number on all the cultivars. Then the number of adults, the number was lowest on cultivars gray and short-in. Yeah, they are short-in here. Then this is gray. They attracted the lowest number of adults. These are eggs again. The egg counts throughout adults. The highest number of eggs we have found on week 2, then tapered down, and there's a little increase during week 5. These are the various varieties, the various cultivars. As the eggs are distributed on them, there's a low number of eggs on early prolific, low number of eggs on gourmet gold hybrid, low number of eggs on the green eclipse. In summarizing, the number of young adults was highest in week 1. The number of young adults was also lowest on cultivars, golden goose hybrid, black beauty, green eclipse, golden glory, gray, and short-in. The number of Mia1 eggs was lowest on lioness, fortune gray, short-in hybrid, early prolific, and gourmet gold hybrid, and then green eclipse. Most productive yellow squash cultivars were the lioness, fortune, amber peak, the gourmet gold glory. Most productive zucchini cultivar was golden glory. Most promising cultivars based on yield attraction, there's a attraction to the whitefly adults, the number of eggs on the various cultivars. Then the most promising cultivars were golden glory, gray, short-in, Fortune, Gourmet Gold, Lioness, and then Green Eclipse. These are our laboratory staff that contributed to this study, and these are some of our students. Again, funding came from USDA ARS. If you have questions, you may ask them. The two studies, his study is different. What we did is this, we started monitoring the sixth week following emergence, right? And then continue to look at the adult populations. It's possible that temperature could play effect, and also rainfall, because we had we had two fall trials, 2021 and 2022. If you look at both, 2021 had higher incidence in our own study than 2022. So as a general rule of thumb, temperature could, climatic conditions could play a role in the number. But if you carry out the study over several seasons, then you can unequivocally say that maybe at this particular season, you're more likely to have whitefly population. But we know that when the conditions are very dry, whitefly populations are more likely to be high. So, and this would vary from season. Even if it is a fall season, over several years, the populations would differ. Because we got very high populations in 2019. We've not been able to find that in subsequent years.
Thank you.
You're very welcome.
Dr. Alvin Simmons with USDA will be talking on USDA Whitefly research update.
Hello. Great. And thanks to all of you who came back from the break. Thank you. And so I will be briefly talking on some things in which you already know, some things in which you may not know, but it will be very brief, and I do have several slides that I wish to go through, but I'll finish on time. And I trust that most, if not all of you, know that the Agricultural Research Service is the primary agency of USDA that does scientific research. And what ARS does is try to find solutions for the agricultural problems. And speaking of problems, we all know that whiteflies are major problems. And unfortunately, it feeds on so many different types of species of plants, over 1,000 different plant species, not to mention the viruses that it transmits. And so the Agricultural Research Service has in-house projects at 5 different locations, Arizona, 5 different laboratories, 5 different states, in Arizona, California, Florida, Georgia, and South Carolina. And I'd like to briefly mention about each of the locations where projects are being done on whiteflies for in-house, starting with the U.S. Arid Land and Agriculture Research Center in Maricopa, Arizona, in which their research is focused on biological control of whiteflies for the most part, looking at the thresholds and using the IPM where the biocontrol fits. And part of the challenge, too, is that with the new BT content, how that impact for the control target for thrips and ligas, how that carry over to whiteflies. Because in cotton, whiteflies are a big driver for the vegetable component. In Arizona, the pink bollworm have required a lot of applications for a given year until it was essentially eradicated in 2006. And then since that time, there were a lot fewer pesticides being needed, insecticides being needed. And as a result, the natural enemies really had a chance to actually, a fighting chance to be able to help in the system. And then I'd like to just briefly transition to California for the Salinas Laboratory, the Crop Improvement and Protection Research Unit. They are focused on biocontrol, biological and biotechnology approaches for the management of insect vectors and vector-borne viruses that are affecting the vegetable crops. Bill Wintermantle and his team have developed 2 methods for detecting mixed infections of whitefly transmitted viruses in cucurbit crops. And of course, in the real world, it's not just one, usually it's not just one whitefly transmitted virus in the system, but multiple ones. And also, Wintermantle and his team are monitoring the continued spread of viruses, which we know viruses continue to spread, not only in California or in Arizona, but also in the Southeast, as well as the whitefly itself. I mean, the vector continues to spread. And as illustrated here, your lower left, the field can be completely infected by whitefly transmitted viruses. They are also working on the development of diagnostics, a tool for the identification of whitefly transmitted viruses. On the one hand, it's fortunately that there are only a few viruses in the U.S. at this time. However, of course, we know that in the future, eventually there will be more. And that also include in the future, it's likely that the tomato virus will show up. And so by having the diagnostic tools, USDA, APHIS, and others will be able to help sample to identify, and so we can intercept, if possible, before these actually get established in the U.S. And something in which, if I don't have any other thing you remember, this is something that you really hope you remember, and that is for the watermelon carotid stunt virus. For the first time this past fall, it was the team detected this virus in melon and watermelon in Arizona and in California. It was previously, we knew that, we in the scientific community knew that it was in the U.S., in wild plants in the desert, cacti, but this is the first time it's being known to be in cucurbit, in crops. Although in Mexico, yes. But like other viruses, the likelihood of it to spread is high. So it's important to watch out for this. Speaking of Salinas, the USDA ARS facility in Salinas is brand new. They had their ribbon cutting back in early November, and this is a state-of-the-art facility for research in the building and in state-of-the-art laboratories, greenhouses. And this is for vegetables and fruits. And in fact, for the ribbon cutting, former congressman from Georgia was one of the guest speakers, Jack Kingston, among others. Now I'd like to move across the country to Florida. The U.S. Horticultural Research Laboratory in Fort Pierce, Florida, has two projects. The first, disease and fruit, vegetables, and ornamentals, is a pathology project. The second, whitefly pest of vegetables and ornamental plants, is an entomologist project, and I'll just focus on briefly on the entomology project. Unfortunately, in Florida, there are over 75 different species of whiteflies. Overall, around the world, there are almost 1,800 species of whiteflies. Fortunately, most of these are of no significance for agriculture. And unfortunately, in Florida, almost every year, there's a new whitefly species that enter. For Dr. Mohammed Hoffman and Cindy McKenzie, they have developed a chemical control plan for the ficus whitefly. This is one of the invasive whitefly species, and in collaboration with some University of Florida colleagues. And this is also called the fig whitefly. They also have integrated natural enemies along with chemical control and designed the first integrated management plan in the U.S. for the ficus whitefly. They also reported a bacteria endosimian that was, for the first time, was found to be transmitted by a parasitic, whitefly parasitic wasp from an infected whitefly to a whitefly with this endosimian versus one without the endosimians. The endosimians is beneficial to the whitefly for it many different things in terms of its ability to feed on the different plants. And so, again, the parasitoid, the wasp, was able to vector the endosimians from whitefly with the endosimian versus the ones which did not have it. And I think this is a really pretty thrips, at least to me it is. And this is for the Franklin thrips vestiformis, also called the vestiform thrip. It's a predator. It feeds on a wide range of different small arthropods. In Europe, it has been, or is being marketed for a biocontrol for plant-feeding thrips. They, Mackenzie and her team were able to demonstrate the feeding capacity of this thrip on whiteflies, which is impressive. It can feed within 24 hours. An adult can feed on about 90 eggs, whitefly eggs, within that period of time, over 20 whitefly nymphs within 24 hours. Plus, the larvae do feed on the plant-feeding thrips likewise. And they have just recently submitted a manuscript for a journal consideration for publication on this. In Gainesville, Florida, for the Center for Medical and Agricultural Veterinary Entomology, they are conducting research on biological-based tactics, and including the push-pull technology. And shown here is Dr. Susie Legaspi. She had pretty much most of her career on whiteflies and biocontrol. This past fall, she retired. And so, but they're still doing research on the push-pull within that unit. And this illustrates the technology of having plants which would repel the insects, in this case whiteflies, within the target crop and push them to, or rather pull them to other plants that will pull them as a trap for away from the target crop. And now let's move up to Georgia, in which there are 2 locations that have in-house for ARS whitefly research. And one is at the Fruit and Tree Nut Research Unit in Byron, Georgia. And that work on microbial control being led by Dr. David Shapiro-Elan. And he's conducting the research in collaboration with University of Georgia, Mike Tays, with some of the support personnel, including Dr. Xiaowei Wu, who just recently joined the Ohio State University as a faculty position there. And then, fortunately, we had Dr. Jermaine Pierre to join as a postdoc, who is physically assisting David Shapiro-Elan, plus also research support from Catherine Anderson and Ashley Algray, who are University of Georgia employees. They are working on a number of different strains of fungi, but these fungi within 3 to 7 days can actually kill the whiteflies. The other location in Georgia with in-house whitefly research is relatively new, just a couple years now, and that's in Tifton, Georgia, for the Southeast Watershed Research Unit. Research being conducted by Jonathan O'Hearn on pests in the landscape. And among these pests are whiteflies, in which he's looking at environmental factors on the whiteflies populations, and also how different environmental factors, such as the wind, would affect the movement of whiteflies for shorter distance and a little bit longer distance. Additional work which he's doing is on chemical and biocontrol with mites against whiteflies, looking at different species of the mites and also different biorational compounds. He further looking at an overwintering shelter in the field or on the field edges to help support the lady beetle Harmonia, which can be a problem to get into people's homes to seek shelter. How in this case, he's trying to provide shelter for them so that when the spring kick around, they are there to be able and feel already to conveniently do what they do best, and that is being a predator. And also, he's conducting some research on the ultraviolet C rays, the light to kill whiteflies, in which he's seen in preliminary studies of the eggs having success of killing eggs of the whiteflies. And I'd like to just briefly talk about some of the ARS in-house whitefly research in Charleston, South Carolina. There are 3 projects. 2 are pathology projects. One of those genetic and genomic-based disease management. The other one, disease in vegetable crops. Those are both pathology ones. And I will be focused on the entomology, since I'm an entomologist, the entomology project, and that's on pest management in vegetable crops. And within this project, we also have some funding support for a whitefly management team, which includes University of Georgia, Fort Valley State University, and Auburn University. And this is to provide solutions for whitefly management in vegetable crops in the southeastern U.S. as a focus. But I will not be focusing on that component. I'd like to just mention for Dr. Sharon Andreessen, who is a vector entomologist, working with whitefly transmitted viruses management, including for different species of viruses, the endosymbionts, transgenic tomatoes as a tool for whitefly mortality, and also on the biological control of the whiteflies. And this is just illustrate on targeting, looking at the upregulated and the downregulated genomics component of the whiteflies in the system. For the whitefly endosymbionts, one of the questions is, do certain symbionts, bacteria, affect virus transmission? And also a part of that too is, if so, if we could target these bacteria to basically interrupt the transmission of the virus. And that's works that's ongoing, as well as the taxonomy of Erythromocerus, postdoc Zach Leahy, because of the expertise is working in this area, because the taxonomy is pretty messy right now, and which needs some attention on that. To switch to a different pest, and that is the guava root knot nematodes, as illustrated in this map on the distribution. It initially in Florida continued to spread, and it continued to spread. And so we know in the real world, whiteflies are not in the field by itself without there being any other pests or disease, but there are other things going on. And so my colleague, Dr. Will Rutter, had detected some resistance to the root knot nematode to a line of peppers. And we further went to look and see if there may also be some resistance to whiteflies. And the answer is yes. We're seeing some resistance in terms of the performance of the whiteflies on the plant, in terms of the attraction of the whiteflies to the plants, and the feeding of the whiteflies on the plant. And for the Agricultural Research Service conducts research to support pesticide registration on specialty crops through the Hour 4 program. And this includes, continually, there are products against whiteflies. And these different pesticides, and these are all sorts of pesticides which are being looked at, including in fruits, vegetables, nuts, floral crops, and others. From the research in which USDA ARS does, the field, the laboratory aspects of this in support of the IR4 has an annual impact of $1.3 billion. And with that, I'd like to just acknowledge collectively the ARS researchers in Arizona, California, Florida, Georgia, and South Carolina, the Whitefly team collaborators, ARS, University of Georgia, Fort Valley State University, Auburn University, and others. And with that, I believe I have time for questions. Okay. So the resistance, twofold, could be twofold. One, that could be the resistance to pesticides. There could be resistance to plants. In some cases, those resistance may be related. And your question is, how can those be taken into consideration? Those are questions that are being looked at, including University of Georgia are looking at it in terms of resistance to the insecticides. And when we say resistant to the insecticide, that's only part of that resistant to chemicals, because in agricultural systems, there are more than just pesticides that are in the system. For example, for the fertilizers, we do know that different fertilizers, types of fertilizer, can impact the populations on the whiteflies, whether there is some resistance going on, don't know, in that particular case. All right, thank you.
Thank you, Dr. Simmons.
Sure.
For our next presentation, Dr. Andre Da Silva will be talking with an update on Alabama whitefly research.
I don't know why they give my title Update on Alabama. I'm going to give an update in general about what we have been done. Yes, that's what we have been done in Alabama since I moved to Georgia, where we are focused trying to solve our problem with the whitefly in Alabama. Most of our research, not most, but some of our research has been replicated. So last year, you guys saw that I present some of the results that we did in Alabama and Georgia. There are some of the trials that are also being done in Florida. As you guys know, whiteflies is a problem in the entire Southeast. It's not something just located in the Southwest Georgia, but it's also in the South, in the entire South of Alabama. Florida, as you guys know, whitefly are on the entire state. So we are trying to come with some solution for our growers. This year was a little bit different than last year. Every year is different than the other years. We have seen several different whitefly movements. Like Alvin said, there are some predator options. We have seen a different migratory movement of those whiteflies most recently. Last year was different. This year, as I was talking with Storm, Alton Sparks, and here in Georgia, they also had a different movement. Last year, what we noticed is that Alabama was 4 weeks ahead from Georgia. So for us, was whatever was happening in Georgia, 2 weeks. Whatever was happening in Georgia, 2 weeks later was happening in Alabama. That was nice. This year was a little bit different. Georgia saw mild pressure on whitefly, while we didn't see much pressure in early season. Then they got disease, we got a pressure, and we didn't see disease. So that was different, but we had a high pressure. So that's what I want to update you guys. Like, we had the pressure. That pressure for us in Alabama was reported in some area of the Southeast and some area in the Southwest as well. If you talk with like Jake there, like you guys can see that they have a lot of greenhouse, and greenhouse that was a big problem. Like Jake was just mentioned to me, like If you go there and shake some leaves, you would see whiteflies flying everywhere. And for us here in the vegetable industry was also a lot of pressure, but we didn't see disease. Mostly happens what's happened in every other states in the southeast. They migrated from one crop to another, and as we get a heavy rain, they are knocked off, and as we have the cotton being defoliated, they come to our vegetable crops. This year, similar to last year, we did some of insecticide efficacy trials. We had some of new products compared to last year. Last year, results were very similar to what's happened in Georgia. This year, we did not replicate this trial in Georgia, so I would just be showing you guys results from Alabama. But it was not much different from last year. I will not get into details about the products, the rates, but I want you guys just to know that here in this graphic, we have the number of whiteflies in the left graphic, and in your right graphic, we have the number of nymphs that we observed. So as you can see, the untreated treatment where we just sprayed water, like we just had like the highest whitefly number of adults per leaf, while we have the lowest, but we have the lowest number of nymphs. That was like interesting for us, but it's still, the best products were the one in green, X-Real and Savanto rotation were the best products for us. But then this product, RM, the 1963, was like also performed as good. And they were able to also reduce the number of nymphs. So those were our best products. So our current growers' recommendations that we are doing, and I know that in Georgia they are doing the same, probably North Florida, they are very similar. The rotation of X-Real with Savanto or a combination with those two are our best products so far. Dr. Sparks might have already talked about some of those recommendations, but yes, those are our best products so far that we have. The recommendation is still what we growers are using. Just put it here to emphasize what were our best products, and that consequently increased our yield. The yield here is in kilograms per hectare. I don't want you guys to focus on number of yields. Each state has different yields. Each grower has different yields. We want to target our yield, but for us, that was our best yield for in yellow squash, in the yellow squash crop. You can see that the same results replicated here responded to yield. So saying that the whitefly adults as well as the nymph make an impact, but adults seems to be more impactful for us. Okay, so this is basically what we are seeing as far as insecticides. We have always recommended X-Real, Savanto, and NAC. Those are our best options so far, and I don't think we're going to update our recommendations so soon unless we have a very strong product. Moving gears a little bit about how we can apply those products, I would like to talk really quick about what we are introducing as new technology in our research trials, but also extension trials, is the use of drones. And I'm not talking about aerial drone. We are also using terrestrial drone. So this year we did a trial with the DJI T40 before you guys asking me how much it would cost for the grower. So the starting point would be $30,000. If you want to buy attachments, that's going to increase the cost. Okay, but that's the base one. That's what we have is a DJI T40. As you can see, we could be able to fly it on top of our crop when it was in the flowering stage, and we could apply some products to test it. So we compare those drones with the regular boom sprayer application. So here you can see we did an application in one of our fields, and we also use a terrestrial drone. That's a new one. It's the R150. It's an X-Ag R150. We also use it to spray. So basically, this sprayer here is an automated sprayer as well as the drone. It's all, you can program your flying or your terrestrial movement. This one particularly grow between beds, and you have air blasters who will spray the crop up and down, so it minimizes the labor that you have, and we compare those ones with the regular drone. I tried to find one of my videos spraying it, but I didn't find, but as you can see in this video, quick video here, it just throw a lot of water. So it's not simulate, but it's basically is an air blaster that you can spray the crop. The only point if you are a grower and you're getting this, it's because the way how these particular R150 was developed, and that's the only one that we have so far, is the tire size, them are a little bit bigger than our regular bed size. So if you are a grower, we use a 6-foot center to center bed. That gives you that 3-foot tire track in between. And this one has a 5-foot bed. So if you set up a bed at 5 feet, you're gonna have a problem. So keep that in mind if you are planning to buy one of those. I'm not saying anybody to try to buy it, but you're gonna need to adjust your bed spacing. Otherwise, you're gonna have problems with that because you're gonna drive your drone or your rover, as we call it, on top of the bed, and you don't wanna do that, okay? So, and the cost is estimated the initial one is around $35,000, and as you buy attachment, you're gonna have increase in cost. Just to let you know, one thing that I forgot to tell you guys about the drone is that this drone here, it's able not only to spray insecticide or herbicides or any other fungicide, but it's also able to spray fertilizer. It's also to spray a granular fertilizer, liquid fertilizer, and you can use it to plant a cover crop and broadcast seeds. So it's very beneficial if you're thinking about it. It's not only one spraying thing, you can do more. So when we compare those two applied during the season on a tomato plant, so there we have a tomato plant and we compare it with the tractor regular boom application. We introduced it, we just would like to see in the initial level is to see the uniformity of application of the products, since we already showed the efficacy of those products. But to see the uniformity of application, we introduced yellow cards that we could see on the top leaf, on the middle leaf, and then on the bottom leaf. And we analyzed how well those products are distributing the products all the way through the plant. And as you can see here, we have the blue card. Those blue cards, as blue it is, as the water hit the cards or hit the leaves. So as you can see, all products perform pretty well. The air blaster has more smaller droplets, while the T40 and the tractor were very similar. As you go down, the air blaster one or the terrestrial one performed a little bit better in the bottom of the plant. And that's because it's not applying from the top, but it's applying the entire plant. While the other ones were applying the top, the boom and the drone. So that's like a benefit of the air blasters. But like I said before, it just spray or do some foliar fertilizer application. But as you can see, those technologies can increase the benefit, can increase the efficacy of application and reduce the labor, what we're gonna talk in the next slides. I will not be showing any percentage of cover. We are still analyzing part of the data. I'm not showing any whitefly control yet. When we apply that, we have like not much whitefly as we would like to, but at least we can see the uniformity of application, and so that those new technology, yes, can be used for spraying in our vegetable crops. There is not much. There's a lot in row crop, but not much in vegetable crops. So just to see what is the pro and cons of those precision agricultural tools is, yes, it's a precision application. You can focus and target only your row. You don't necessarily need to do like the entire field. You can just apply low, if you're flying your field, you can low your drone and apply only in that row. We tried to do in 3 rows, but it's did not work very well, so we decided to keep it only on single rows. It's improved the uniformity or equal the uniformity as a boom sprayer, and it saved on product and labor because when we did that, we were able to spray relatively only in the top of the bed with the drone and exactly in the bed with the air blaster, so we could reduce a little bit our use of product. The only cons that we have found so far, as this is still a testing, is we are very battery dependent. Those runs on battery, so you need to have a station on field if you have a large field. So just thinking about that those batteries can run like for 1 hour, for 30 minutes. So you need to have like extra battery And usually we recommend to have a charger there where you're going to be charging one battery while the other one is working. That's doable, and that's what usually row crop guys are doing. They have their own station where the drone can fly, spray, come back by itself, sit in the station, and you just change the battery. It's auto refill for the drone, and you can do that. But to do that, as I said, you need specific training. As your tractor driver will need a specific training. So it's not much different, but it's a specific training. You need to be attention if you are spraying next to airports, because you're going to need a lot of permission. Trust me, a lot of regulations. But it's doable. And, but one thing that we notice, it's much more automated. And you don't need to have only one doing the job. If you have a large field, you can have multiple drones doing the job. Same thing for not only for the T40, but also for the R150. Like, this R150 amazed me because it sprayed, and if you have a deer or a dog crossing they have a camera in front of it and recognize it and send a sign to your cell phone saying, we identify an animal or something passing by. And it stops. Then you go there and check what it is, or you can look through the camera and say, walk again, and it will keep you walking. It go all the way to the field, turn, and come back, all automated. So you guys are able to do that. You just need to do your fly plan, as we call. And we say fly plan, but it's for a rover. So, and one thing that we don't know yet is the lifespan of those equipment. Since it's new technology, it's just coming, so we don't know how long it's gonna last. What we can know is technology is going so fast that it's probably gonna not be updated by the time it fades out. So maybe there will be next, like the T40 was released December 2022. This R150 was released in February last year. They might be releasing a new one this year. And those products are still good, but they are not a new technology. So technology is going so fast that you need to keep in mind that you might not have the most updated equipment, but doesn't mean that your equipment is not good to do the work. Okay, so we don't know the lifespan of those equipment yet. Switching gears about a little bit of research, not recommendation for the growers, although it's important here. That's probably what Bill is most interested for. Is about our tomato trials that we have. If you were here last year, and we have communicated, we are doing like several trials on tomatoes to see the resistance for whiteflies, in whiteflies. We are using wild material, so this is not commercial material. It's not available for growers, although we test some of the commercial materials. But it's just, what we are trying to do is identify what creates resistance to whiteflies on tomatoes, or what is there on tomatoes that give resistance to whitefly. So last year we have tried several wild materials. This year we removed some of the materials that did not perform well, and we added new ones. So the hybrochides one were the new one that we introduced, some of Galapagos as one, and we kept some of the ones that performed the best, like the G2925A there in our trials. And what we're going to go is just quickly, since this is more an extension meeting, or this is more a growers meeting, I don't want to go into research, so I'm going to go quickly through the research so those who are here more interested in research can also get some information. So on whitefly population, on those trials we planted, we measured the whiteflies, we controlled the whitefly population, and the number of whitefly eggs in the top left, in the top right is the number of nymphs, and in the bottom here is the number of adults. As you can see, last year we have some significant difference in those, but this year the only significant difference we saw among those cultivars or those materials sessions were for the number of nymphs. And you can see this material here, the 2925, which was one of our best last year, were not our best this year. So we had new materials that we introduced that were better than before. So we know that there are new species, or there are species that has even more resistance or can create resistance to whiteflies. But this was particular to nymphs. We didn't see difference for adults, neither the number of eggs. And this, as we guys saw last year, was related to the trichomes. That's what we are associating, and that's what is in the literature. Because trichomes, we have the glandular ones and the non-glandular ones. The glandular ones are the ones who produce volatiles, which are metabolics or are chemicals that can repel the whiteflies. While the glandular ones that create, the non-glandular ones, which are the hairy ones, when you grab a tomato leaf and you see that hair tomato, that's a non-glandular trichome. They don't produce volatiles. So you want to stick with the ones that produce volatiles because they will be creating the resistance. So just for you guys to see, in the bottom here we have commercial varieties. In the top here we have non-commercial wild material. You see the big ones. We have several big ones on the top, while here we don't have a single one. When we measure and count those numbers in our leaves, you can see the total number of trichomes. Commercial materials, yes, does have more trichomes in general. That's the total number on the top graphic. While in the bottom left graph and the bottom right graph, on the left one, we have the non-glandular, so they are just the hair one. They don't produce volatiles. While in the left, your left one, we have the ones that are glandular and produce volatiles. It's much larger. Once again, when we come back to results from last year, the G2925-8 is the one that produced the best last year. This year, it was all in the bottom. So we are seeing new materials that have that ability. So we are trying to investigate it more. Why are they doing? So this year, different than last year when we just were able to say there was or there were not, like we were able to better identify. I will not get into this graphic with you guys. It's a little bit complicated. It's much more research, but I just want to show you guys this. As light it is, the bar, more volatile that in the literature says that it's repel whitefly, that particular cultivar has. And this particular material, the one that ends in 7826, is the one who showed the lightest one. Those are Z-gibberine materials, and that's all the type of volatiles or chemical profiles that we want in our tomato. So as you can see, it's much better than the materials that we have in the previous years. So this material here and this material here is the one that are showing promise to us. We're going to continue repeating this trial and work with breeders to introduce that to new cultivars, but so far that's what we have been seeing in our tomato trials. So just to see you, we did a little bit more in-depth statistical analysis. We did a PCA, or we call a principal component analysis. I will try to explain it pretty simple. We have three groups that were identified, that particular variety, that material that produce a lot of volatile, the commercial material, and all other materials here, separated. As far as as closer as they are, more similarity they have. So that one have like a different aspect from the other ones. And we saw that glandular, as glandular it is, our trichomes, as more glandular trichomes we have, less adult whitefly we have. So my question was, like for growers, like how hairy should be our tomatoes? We know that cotton, as more hairy is your cotton, more whitefly is attracted. Correct, Storm? What about tomatoes? Should our tomatoes be hairy or should they don't be hairy to attract those whiteflies? What should our growers be looking for in their cultivars? That's the kind of things that we are trying to identify right now. And just to finalize and conclude a little bit my talk, and for the growers, I would like to say strategically plant vegetable crops, time is key. Like I said in the beginning, last year we were in Alabama, were 2 weeks ahead from Georgia. So when Storm had a problem here, I wish he called me and say, hey, I had a problem, so I'm gonna have a problem there. But I need to keep bugging him, hey, what do you have there? What do you have there? Or he wanna talk about soccer, and then I say, hey, Brazil again. Okay, so keeping that in mind, plan ahead. X-Real and Savanto are still our best options as we have And I believe you already talked something about that storm on the, but for us, X-Rio and Savanto and NAC are our best options. I just would like to remind you guys, we didn't do any row cover or silver plastic trial this year, but what I would like to say is yes, for larger growers, as we were talking with Mr. Bilbrim, it's labor intensive for laying a cover is the problem. But for small growers, that can be a good option. So if you are a small grower, putting a row cover can save you some money on products. And remove that cover at flowering. Timing of cover remover is key. If you're planting yellow squash or zucchini, about 18 days is the time that you want to remove that cover. Laying a silver plastic is a good strategy to show it. For us, I know we don't have much pressure in Alabama, in North Alabama, for whiteflies as we have it in the South. In the South is our worst case scenario. But I have seen growers in North Alabama like laying 200 acres of silver plastic for tomato. So because it's not beneficial only for whitefly, but other insects. So that silver plastic is important. For the researchers, I just listed here what are our sessions that are performing the best and showing some promising effects against whitefly nymphs. Also, number of those same ones present a higher number of glandular trichomes, and the metabolites that we are looking for is the gingerbread, and then I will not mention those names here because it's there is no need, but this PI here, the 7826, were the one that performed the best in our trials. With that, I would like to thank the USDA and the IR4 because they are the ones who funded us, as this entire section was talking about whiteflies, and I'm open for questions. Yes?
So for I just want to understand clearly, pretty much your wild tomatoes, the ones that did the best, and they weren't so hairy, are they like the potato leaf type of, and determinate type of tomatoes?
So those are wild material. So we have different types of materials there. So they are not, I'm not saying that they weren't performed the best. They are the one who had lower in this year, they are the one who had the lower number of whiteflies compared to those 4 commercial material. They had more glandular trichomes, while the commercial material, they have more non-glandular trichomes. Non-glandular trichomes, they look like that spear, while glandular ones look like that small round ball that they have on it. That's where they keep their chemical profile, their metabolites. So what is it? That's where it is produced. So that metabolites repel the whitefly, while the other one doesn't have it. So that's why I'm saying the other ones are hairy because they have more non-glandular ones. But doesn't mean that like what we have been trying, last year what's happened is our commercial material were full of whiteflies, but no nymphs and no eggs. While this year there was some nymphs and eggs there, but less adults whitefly, no difference significant from the other one. Our population this year was a little bit lower than last year. So we are still trying, like we know, it's known that the glandules of those trichomes, they produce metabolites that will repel whiteflies. So what we are trying to is to identify those who has the most. What do you mean?
You said that they have the glandular materials that repel the whitefly. Is it possible to extract that basic material, that metabolic material, to synthesize it
And use it as a insecticide? Yes, it's possible, but that would be more like, it's a different project. Yeah, it's an additional project. Yes, I would love to. Like, we can create a product. We can create an insecticide. That would be awesome. Yes, Bill.
So were you able to monitor the amount of gingerbread or whatever it is you're looking for in there to see if there was a difference between last year and this year? Would that have any influence? And if there was, do you have any idea why there was?
So The difference between, you mean,
Last year's results pointed in a certain direction, this year's results were a little different. Was there a difference in the amount that was being produced by the size of the trichomes from year to year? And if there was, do you know why?
yeah, so that's that's a great question. So basically what he's asking, I would just try to explain to the growers that are in the room so they know what we're talking about. So he's asking if there was difference from last year in the amount of those metabolites last year compared to these years. Unfortunately, Dr. Rodriguez that is in the back, she's laughing because of the question, because she's helping me on that analysis. And just for you guys to know, she's my lovely wife as well. So, but yeah, the problem is last year, the methodology that we used to extract that, I'm not completely trusting that because it's a new technique. So I don't want to say that there were differences. So this year we give a step back on the new tech in the methodology, and we use the traditional methodology to do it, and we were able to quantify it. So I'm not able to say if weather, for example, is impact the production of that metabolite. Next year, I will be able to tell you that, but this year, unfortunately, I can't. But we use two different methodologies in both years. And we saw that last year methodology was not as accurate as this year, although it's a much faster methodology. Yes?
So you mentioned that you had multiple acquisitions and you checked their biochemical profiling for the specific compound. Did you also check like number of trichomes versus the level of a specific biochemical compound?
We also measure the number of trichomes, granular trichomes and non-granular trichomes as well in all those accessions.
And did you see any correlation between the granular versus non-granular for the presence of specific chemicals?
So we are doing that analysis for but yes, we are seeing some, we did some correlation. When as higher is the number of glandular trichomes, the higher will be the number of those chemicals. Any more questions? If you want to talk later, I will be in the room. You guys are more than welcome to talk with me. Thank you, guys. I appreciate it.
That was our last presentation for the session. Thank you all for attending.